All right, today we're going to cover genetics, Mendelian genetics. So Mendelian genetics was discovered by a guy named Gregor Mendel, who was an Austrian monk. He used pea plants to figure out how things are inherited, how genes and that kind of stuff are passed on. So from meiosis, you remember that you get one copy from mom and one copy from dad. So let's just say this is a chromosome from mom, and then here's a chromosome from dad. So if these were the same chromosomes or homologous chromosomes, we would see that they would code for the same stuff, meaning they would have the same genes. So what is a gene? A gene is a section of DNA that codes for some type of trait. So it is a DNA that codes for a trait, a physical trait of some sort. So let's say, and so remember, the trait is comprised of two copies, one from mom and one from dad. So let's just say mom gave you, and we'll just use eye color. So for now, let's just say, and we'll talk about this later, but let's just say big B is for brown eyes and little b is for blue eyes. So this is for eye color. So let's say mom gave you big B and dad gave you little b. Okay. All right. So an allele is one of the copies. So it's a copy of a gene. And so you have two copies, one from mom and one from dad, right? So in this case, in our example, the alleles are big B and little b. Those are your two alleles. And so the gene is basically the two alleles. So the combination of those two alleles is what we call a gene. And then dominant is, so a dominant allele, I should put allele, a dominant allele is the stronger allele. And we'll talk about what stronger means. And so in this case, it's always capital, so big B. And the recessive allele is the weaker allele. And so we use lowercase, so it's a lowercase b. All right, so you have two alleles, one from mom, one from dad, that form a gene. And the allele can either be dominant, which we represent as a capital letter, or recessive, lowercase. All right, so what is a genotype? The genotype is made up of your two alleles. So, for example, since you had big B, little b, that's the genotype. So that's what's on your genes, right? And so there's three types of genotypes. You could be heterozygous. So the term hetero means different, right? So hetero means different. So big B, little b. So that means you have two different alleles. So you have a dominant and a recessive. So big B, little b. And then you could be homozygous. But there's two types of homozygous. So the prefix homo means the same. So you could be homozygous dominant or homozygous recessive. So here you could be big B, dominant, or little b, right? So both parents gave you both recessive alleles, or both parents gave you two dominant alleles. The phenotype is your physical trait. So let's just remind ourselves that big B is brown, little b is blue. So in this example, your phenotype is a physical trait. So for example, let's just take, if you're heterozygous, then your phenotype would be, well, you have a dominant or recessive. Since the dominant is stronger, your phenotype would be brown, right? So this would be your genotype, and this would be your phenotype. And then if you had big B, you're homozygous dominant, so your eye color would be brown. And then big B, oops, sorry, your homozygous recessive, it would be blue. Okay? All right. All right, so Mendel came up with these ideas. And so there's 3 principles that you have to know. The principle of dominance, principle of segregation, and the principle of independent assortment. So the principle of dominance is just what we talked about. One allele is dominant to the recessive. So, and the dominant is expressed phenotypically. So remember, in the heterozygous case, you have big B, little b, it's going to be brown, right? Why? Because one allele is more dominant over the other. So actually, this is kind of interesting. So here's the parental cross. So you have a yellow seed crossed with a green seed. And so all the kids are yellow. So which do you think is dominant? Well, since all the kids are yellow, that means that the dominant allele is yellow. So this is actually how Mendel figured it out. So what's the possible genotype of the yellow seed? So if we know that yellow is now dominant to green, so we can now say that yellow is going to be big Y, capital Y, and then green is little y. Oops. So what is the possible genotype of the parental yellow? Well, it could be homozygous dominant, or it could be heterozygous, right? What is the genotype of the green seed? Well, it has to be both recessive, because that's the only way green can be shown. And then what is the genotype of the F1 seed? Well, it could be big Y, or it could be big Y, little y. So it could be homozygous dominant or heterozygous. Now, if you think about it, it has to be what, though? It has to be homozygous dominant. Or, I'm sorry, heterozygous. Okay? All right. So, principal segregation. So your mom and dad gave you one allele each. So you got one copy from mom, one copy from dad. And that's because in meiosis you get haploids with only one copy, right? Instead of 2 copies, it's not diploid. And then the last one is the principle of independent assortment. So it basically says that all traits or all genes assort independently. So what that means is, for example, if you have like eye color is going to be different than your hair color, and so they won't, like, if you have brown eyes, you're not going to have brown hair. They don't always go with each other. They just kind of independently do their own thing, so they're not dependent on each other. Okay. So how do we figure out, like, what an offspring might be, or the genotype or phenotype of an offspring? So we use what's called a Punnett square. So a Punnett square is used to predict the offspring. So let's just do an example. So it says 2 heterozygous purple plants are crossed. So we know that purple is dominant and white is recessive. So big P is purple and little p is white. So if there's 2 heterozygous, it's going to look like this. So this is what we call the cross. And so we're going to go ahead and make a Punnett square here. And so here, it doesn't matter what side mom or dad goes on, but one parent is going to go on the top and one parent goes on the bottom. So if the parent has heterozygous, they can either give their offspring a big P or a little p. So these are the two possibilities, and then it could give this, right? So we're basically figure out what are the combinations that they can give them. So in this case, what we do is we can put this here, and then here, and this could be here, like that, and then here. So basically, we're figuring out all the possible combinations. Okay? And then from this, we can figure out the genotypic ratio. And so, let's see. So, 1 out of the 4 possibilities, out of the 4, or 25%, is going to be homozygous dominant. 2 out of the 4 will be heterozygous, and then 1 out of the 4 will be homozygous recessive. Okay, what about the phenotypic ratio? Well, these, because they contain at least one dominant allele, 75% will be purple, and then 25%, or 1 out of 4, will be white. All right, so try the next one on your own. I'll just do the example. So we have a heterozygous and homozygous dominant. So we have big P, little p, like that, right? And so now we're going to create a Punnett square. So what you'll see is 2 out of 4 is little p, little p. 2 out of 4 is big P, or heterozygous. So 50% and 50%. And then you also know that 50% will be white and 50% will be purple. Okay.